In language now. Good morning, everyone. It's a pleasure to have with us today Mauricio Girardi-Schappo, Professor Mauricio Girardi-Schappo. Mauricio holds a PhD in physics from the Federal University of Santa Catarina, UFSC, in Florianópolis, Brazil, specializing in statistical mechanics and critical phenomena. He completed postdoctoral research at McGill University in Canada, the University of São Paulo in Brazil, and the University of Ottawa in Canada. He's the head of the Neurophysics Lab at the Physics Department of UFSC, currently funded by FAPESC, which is the Santa Catarina Science Support Foundation, and by a CNPq fellowship. In his lab, they are building robots to emulate animal navigation, as well as developing stochastic models of the behavior and brain circuits involved in spatial memory formation and epilepsy. He's also a skydiver in his spare time. So it's a pleasure to have Mauricio here. He'll talk about the title. You can see the title. So without further ado, let's start.
Okay, thank you. Thank you, Rocky, for the kind introduction. I hope you guys can understand my English. And just because I'm giving the seminar, I just started listening to some hammer knocking on the other wall. I don't know if you can hear it. So sorry if you have any noise. Let me know. You can stop me at any time with questions. Okay, so I'm going to talk about a little bit neural adaptation and spatial learning. This work is something that I have developed started developing in my previous postdoc at the University of Ottawa under the supervision of Andre Lontin and Leonard Miller. So these are really, really nice guys. I still keep collaborations with them. And also part of the works that I'm going to be talking about today were developed as part of PhD studies of both Joan Shu, which is, we call her Kelly. That's the name that she assumed, to stop people from mispronouncing her Chinese name, and also Antoine Trim. Okay, so these are, hold on, okay, right. So I'm going to talk a little bit about the dentate gyrus and the things that happen. The dentate gyrus are, place cells that are there to code for spatial positions. The dentate gyrus is also responsible for pattern separation and memory and learning. Okay, so I split this conversation we're going to have in 3 stages. The first, I'm going to talk a little bit about the behavior scale, which is an experiment that we did in the University of Ottawa. And then I'm going to talk a little bit about the cellular scale, which is another experiment that we did in the University of Ottawa to be able to extract some features from the membrane potential of mossy cells. And also, I'm going to talk a little bit about modeling and the neuropopulation scale, which is something that I have been developing to study the coding of the networks. To start with, I'm going to talk about this experiment. I'm going to just show you a video. It's a very simple experiment. I'm going to give you more details as the talk progresses. So the experiment is just basically a hidden food in the arena, and the mouse has to find the food. I'm going to give you more details. I'm just going to show you this quick video. So now the mouse is looking for the food. The food is one of these holes in the ground, on the ground. And in this particular case, the mouse has this wire coming out of its brain because they were recording the activity of the brain. But in the experiments that I'm going to talk to you about, we didn't record brain activity. So this is like a 2.0 version of the experiment. It's searching, it's probing, it's foraging, it's hungry, so it wants to find food. And then, almost there, boom, it found the food, it eats, and then it comes back home. So it's quite simple experiment. Okay, so Kelly was the one who proposed these experiments initially, a few years ago already, during her master's. And we want to answer 2 questions. And the first one is, can mice navigate using only path integration? Because usually the most common hypothesis is that mice use landmarks, and they really like landmarks to navigate this kind of more open-ish environment. I put here closed but wide environment because it's not exactly like, this kind of T maze or whatever, where the environment is really closed. This is kind of an open environment, but also, you know, it's restricted. And so this is the first question. The second question is, given that they use path integration, which is something I'm going to show you, can they build a cognitive map? Because also the common hypothesis, or actually the kind of assumed to be true, is that mice cannot have cognitive maps. So it was something that was seen in primates, but was not seen outside of these biological, you know, of these kind of animals or primates. I'm not gonna say like any biology keyword here because I'm not sure about them. But anyways, so there are different definitions for cognitive map. We assume this definition by O'Keefe, which is like a flexible navigation or the ability to take some path that was not learned before, okay? So that's the definition of cognitive map that I'm gonna use. So the experiment we have this arena which has 100 holes in the ground. The holes are kind of randomly distributed. The arena has 1.2 meters in diameter, so the mouse is quite small compared to the diameter of the arena. We want to minimize stress so we don't directly manipulate mice. And you know, the arena is built here. You can see on the top. I don't know if you can see my cursor, but hopefully so. So here we kind of have this randomly distributed holes in the first quadrant, and then you take this distribution of holes and you project them rotating to the second quadrant and the third quadrant and then fourth quadrant, so that every hole in the arena has kind of this equivalent position in every quadrant, right? So for example, if the mouse enters from the second quadrant here and it wants to find the food here, the target, which is something that it did in the experiment I just showed you, what is going to happen is if it enters from the third quadrant, it has this kind of equivalent position here in this, you know, rotationally equivalent position, REL, in the second quadrant, which would be the same kind of relative position to the entrance as the target is to the entrance that was trained to the mouse. Okay, so the point being, every hole in the arena has this rotationally equivalent position so that the mouse would be able to go to that position and find food there relative to the entrance it took. And this is important because it allows us to kind of test for the path integration versus the use of landmarks, right? So all these displacements, as you can see, are kind of similar, but starting from different quadrants, right? The target vector or the REL vectors here. Say if you want to go 70 centimeters forward and then 30 centimeters left, that's the same displacement you're going to have from each entrance to find the kind of rotationally equivalent position of the target, REL, okay? Right. So I'm going to show you three different experimental protocols rather. One is the random entrance experiment and then the static entrance experiment. And this set of experiments allows us to test for the use of landmarks versus path integration. I'm going to give you more details on the go. And the third experiment is going to be the two food location experiment, which we've designed to test for the hypothesis of cognitive map. Okay. Right. So first, the random entrance experiment. I'm not measuring time, but you can interrupt me, you know, if you, Rocky or someone else, if you can let me know when there's like 10 minutes left or something.
I'll let you know when there's 10 minutes to go, okay?
Right. So random entrance experiment is basically this that you're seeing. The target is always linked to this kind of landmark on the wall. So for example, here, the target's next to the X on this wall. And then from every different trial, the mice enter the arena from a different quadrant. So for example, if it takes quadrant 1, it's going to have, you know, this trajectory to find the target, or if it enters from quadrant 4, it's going to have this trajectory to find the target. The important feature is that the target is always linked to this landmark. Okay, so if there's significant improvement on the trajectories or on the trajectories that the mouse are going to take, this is evidence of the use of the landmark that are hanging on the wall, and it corroborates this hypothesis of allostatic navigation instead of egotistical navigation. And Also, other than measuring trajectories, we have this camera on top of the arena that's recording everything. We also do this kind of 2-minute probe to test whether the mouse prefers to stay close to the target or to stay away from the target, right? Just to give you a summary of the results, these 2 examples of trajectories recorded in the first trial for 2 different mice that started, for example, in this entrance. And you can see that they dwell for a long time until they find the food. Okay, this is the first trial. But then you look at the, for example, 14th trial, which is at the end of the training, and you see that there isn't much improvement, at least visually, right? You see the same 2 mice, they're entering now from diverse entrances, but they cannot, I mean, you kind of see that maybe they took a little shorter to find the food, but you know, it's still very much like the first trial. So if you look at the 2-minute probe, the target quadrant is quadrant 1 here next to the X, and you see that the time they spent searching this quadrant is approximately 25%, which is basically random if you compare to the time you'd expect, which is 25% for each quadrant, right? So there's no preference for the target quadrant, even though the food is there and the land. I mean, the food's not there because this probe has no food, but the X is there to kind of mark where the food was. And if you plot the distance that the mice walked to find the food versus the trial, I mean, this is the normalized distance according to the target, so that the optimal trajectory would be 1, right? When both distances match. You can see that over the trials, it kind of does improve a little bit. It decreases a little bit the distance the mice walks, but it doesn't go to the optimal trajectory. And if you estimate the learning trial for the distance, you can see that it's going to be around 26. So at trial 26, it's where you would expect the mice to have this kind of more optimal trajectory. And so it shows a stagnant performance, right? The mice are kind of not learning too much in this scenario. Okay, so testing for path integration now, you'd expect to see, so I'm going to define this kind of what I'm calling active sensing or the hole checking of the arena. We can run an algorithm and detect every time the mouse sticks its nose in the hole to see if there's food there, because you can only sense the food from really near the hole for a bunch of different reasons. I'm not gonna give you all the details. If you are interested, you can ask me. But okay, so the mice sticks their nose in the hole, and we can detect that with a computer algorithm, and we call this a hole check. And we use this as a proxy for active sensing the environment while you're looking for food, right? So one measure of error is how many hole checks you did divided by the trajectory, the length of the trajectory you took, right? So you expect from the path integration perspective that the number of errors is going to increase with time. And this is what you see. We divided the trajectory in 2 halves. So the first half of total time the mice took to find the food, and then the second half of total time the mouse took to find the food. And if you plot that over trials, the number of checks or the error, you're going to see that both are kind of very similar to one another. There's a little bit of trend in the second half for the errors to increase over trials, which is something you'd expect from path integration, but still there's no significant increase. If you look at the R value here, it's really small, right? So not really good. And the P is very small, very big. So the errors kind of stay constant. And then the second thing you'd expect from path integration is if the distance of the checks, the distance of each hole check relative to the target would decrease over time, because it would imply that the mice are kind of learning to check closer to the target. And if you plot these over trials, you see that they're stagnant. So in the first half, in the second half, they're constant, they're far away from the target because they're always like 30 centimeters away from the target. So they're not learning the target as well. The distance of checks is not decreasing as you would expect from learning. So yeah, this speaks against path integration in this scenario. And if you look at the spatial distribution of these hole checks, you're going to see that in the beginning, the mice are very prone to check near the walls. This is a very expected behavior because they want to avoid predators in open spaces. But as the mouse learns, for example, in trial 14th, you see that the most hole checks happen towards the center of the arena, however, still far away from the target. So that's the spatial profile here. Okay. Now for the static entrance experiment, it's completely different, the protocol, because now the mouse always starts from the same entrance, right? So every trial of the training, they're always starting, for example, from Q2, okay? So you have a very fixed position of the target relative to the entrance for every training trial for every animal. So in this example, for example, you have to walk 70 centimeters forward and then 20 centimeters left, and you're going to find the food. And then we can do two types of probes with different animals, of course. And then one type of probe is just this 2-minute dwelling on the arena without food, so that we check what's the preference of the mouse in terms of spatial space in the arena, spatial position. And then the second type of probe we can do is, instead of, so we did the training in Q2, but then at the probe trial, we let the mouse enter, for example, from Q4. And then, depending on the decision the mouse takes, if the mouse decides, for example, to go to the REL location of the target, it's evidence of path integration, because it kind of preferred to go towards, you know, this kind of vector that it learned during the training. Or if it prefers to stick to the wall cue, here, for example, the square, then it's a sign that it's using landmarks. So again, just showing you examples of the trajectories. In the first trial, it's pretty much the same as the random entrance because it's the same experiment. But then in the 14th trial, you see a clear difference between the trajectories of the first and the last trial. And then if you plot the heat map of the dwelling, then you're going to see a clear preference for the quadrant where the target is. And here, for example, 50% of the time, the mouse stayed in the quadrant where the target was found during training. And if you plot now the distance, the normalized distance, you see a clear decrease in distance until actually it reaches the optimal trajectory. And you can estimate by fitting an exponential to that curve there, you can estimate that the learning happened at approximately trial number 6. Okay, so there is an optimal performance of the mouse at the end of the training. Now, if you plot the error distributions, you're going to see, again, you expect the errors to increase over time, and then the distance to decrease over time, I mean over learning. And if you look now at the errors, you're going to see that in the first half, it's the same as before, but in the second half, there's a clearer, it's not, you know, that clear cut, but there is a clearer trend of increasing in the second half. You can see that by the R of this fit, okay? And then for the distance of these hole checks to the food, now you can clearly see that this distance definitely decreases over trials, right? So both speak for path integration, and that's exactly what you expect from path integration. And if you plot now the spatial profile, you're going to see in the beginning the same kind of distribution towards the walls, but in the end, you're going to see the distribution migrating towards the trajectory only, because that's the only place in the arena that mouse is walking, right, near the optimal route. Okay, so that alone is evidence of path integration. And now, further evidence of path integration is this second type of probe, which is we rotate the probe entrance according to, you know, from the training entrance. So for example, here, if the mouse entered from the top during the training, and then you rotate this to the side, to the right side, you see that they go to the REL, and every time they go to the REL, never mind what kind of rotation you make, And never mind if you have wall cues or no wall cues. You see, for example, here on the left, we did experiments with no wall cues, with wall cues, in the dark, and with controlling for odor and every other type of control you wish. We did all the controls possible, I mean, that we could think of, and then the referees also asked. So, and also if you plot the spatial profile of hole checks, you can see that it accumulates near the REL and goes nowhere near the, actual target position for the rotated experiments. So this is, together with what I showed you about the errors, this is very strong evidence for path integration that the mice are using and actually ignoring the landmarks for whatever reason. We're not saying that the mouse or the mice do not use landmarks, but we're saying that here in this particular configuration, they're just using path integration, ignoring landmarks altogether. Right, so far we have seen the learning of distance, but then you can ask, how about the learning of directions? And for that, I'm not going to go into all the details here, but we kind of can overlay this kind of square lattice on top of the arena and create this kind of map for the preferential direction of displacement for the mouse. We call this kind of a step map because it kind of inspired by the random walk, where you have this agent or mouse running the arena and takes a step to the right, to the top, to the bottom, to the left, right? And you can estimate these probabilities of taking a step and then show this kind of map of most probable displacement. Also, we can calculate the significance, but I'm just going to show you how the map looks. And this is what you see in the experiment. So each arrow here is a vector of the most probable displacement from that particular position, right? So it is the gradient of probabilities in that particular position. And then you can filter this for statistical significance of these vectors doing some jackknife sampling. And then this on the right is what you see from the filtered data, meaning you only see here the displacements that are statistically significant. And from there, you can estimate the target estimation vector, which is this pink vector here with some error around it, which is the preferential direction from the whole trajectory that the mice took in their run, right? So, and then the target vector, which is this purple vector here pointing to the target. And we can, for example, calculate the angle difference between the two. And here for random and static entrances, random on the left, you see that in the first trial, the target estimation vector is kind of random. And in the last trial, it's also not pointing to the target. However, on the static entrance protocol, you see that they have this difference in the beginning, but in the end, they almost match almost completely, right? And also you see that the step map here generates this kind of flow towards the target, whereas in the random entrance protocol, there's no flow, no clear flow to the target from the significant displacements here. And then if you plot the target TAV deviation, you see also that this kind of, at the 6th trial, the angle is kind of learned already for the static protocol in blue here, whereas for the random protocol, the angle is never learned over trials. Okay, so as I said, we do that for all sorts of different controls, odor gradients, light conditions, and with or without markings. So far, we've seen that random entrance is not significant. The learning is not significant if there is learning. And then for static entrance, the learning happens through path integration. And then how about the cognitive map? So for that, we have this two-target protocol, which is we first train target number A with the static entrance protocol. And then we remove the food from target A, put the food at target B, and then train target B for another 8 trials, for example. And then you do a probe and ask whether the mice can take the shortcut from B to A. And here it's important to notice that the training of target A happens during like 4 days, and then the training of target B happens for another 2 days or something. So when they do the probe, you have to remember that this is like a long-term memory that they have to remember to be able to kind of return to point A after learning point B. So it's kind of a, latent memory there. If they're able to kind of take this shortcut, it means that they are really remembering something from the old days in the perspective of mice, and they're able to, take the shortcut if they do it, right? So yeah, I'm gonna just show you this kind of video from one of the mice in the probe trial. So this mouse that I'm gonna show you, it's already done training for target A and then have done training for target B. This is a 2-probe trial, so there's no food in the arena. The second probe trial, there's no food in the arena, and the question is, what does the mouse do? Oops. And you can see it's gonna go there. Every circle appearing is like a whole check event. I already showed you this as a GIF before, but still you can see here more slowly what it's doing. And as I already did spoil before, you can see that the mouse is going directly to target A. Of course, since it's a very old memory, it's not necessarily going to remember exactly what was the location of the food, but still, it's pretty much, took the route to target B and then the route to target from B to A. So it's quite clearly, you can see that this mouse remembers both target B and target A because, it could have done other stuff as well. For example, if it's anchoring its knowledge to the entrance, it could have gone B start A or other types of then A start B, or you know what I mean? So take other routes, but we've seen that in the majority of the cases, this is what the mice do. And we can do the same analysis we did before for hold checks and TEV estimation. And you see, training target A, that's what you see. Then training target B, this is what you see, which are exactly the same as we've seen for the static entrance protocol because they're static entrance trainings. And then for the probe, which is the novelty here, you can see that there is a flow, a clear flow from target B to target A. And if you estimate the target, the TEV here, this vector that, it points from B to A, right? And the hold checks also are happening quite impressively only near B and near A. The rest of the arena is kind of clear for hold checks. So from this, we believe it's strong enough evidence to show that the mice have this kind of ability to navigate from B to A, even though they have never learned this path before. And we also did this kind of experiment, this 2-target experiment with rotations. And you see the same thing happening that you saw in the static entrance, which is now, if you train from the top entrance here to target A, and then you train from the top to target B, and then on the probe you rotate, and then the mouse enters from the bottom, you see that they go to the REL of B, and then they go to the REL of A. So they repeat the same thing that they did with static entrance and the same thing that they did with the 2-target entrance when the entrances are rotated. Okay, so far we have seen this kind of adaptive behavior and flexible navigation of mice from the behavior level. And what we want to do now is to try and go a little bit further and see what's happening in the hippocampus, or in the dentate gyrus rather. And for that, we want to, I'm gonna give you some, definitions. And the mouse, for example, here is navigating the arena. And if you stick an electrode in the mouse's brain, in the hippocampus, or rather in the dentate gyrus, and for example, you're recording from these 3 types of cells that are very important. One are the granule cells that I'm representing here in blue, and then the mossy cells in red, and the CA3 cells in green. And you're just recording while the mouse runs around the arena. You're going to see some spikes in each of these cells. And these spikes are going to be anchored to some positions in the arena. And if you plot the positions where the spikes happened, you're going to see this kind of clear place fields. So we call this like the place fields of each neuron you're recording from, right? So for example, every time the mouse passes through this red region here, you see a flash. Oops, I think it's changed. Yeah, I changed the colors. So for the place fields in blue, it's the red that's getting activated. So I'm sorry for that. For the place fields in red is the blue that's getting activated. The red's correct, the green's correct. So forgive me for that. But still, the place field is the region of space where that particular neuron is activating over time. So where is the hippocampus? Just to give you, a more concrete picture, if you take this slice of the mouse's brain and then you look at the hippocampus and you zoom in the hippocampus, you see this kind of structure, and the hilus is this kind of small part here that contains this some granule cells and some mossy cells and some CA3 cells. So for example, people have done this experiment. We're doing it right now with these experiments that I told you about. But for example, I'm taking here the paper by Goodsmith, and you can see here the tip of the electrode that they stick in the hippocampus. And they did the experiment. For example, here's the CA3 region, which is this thin layer of cells coming out here. And for example, for this particular neuron, it only activated in this spatial position. So this is the place field for this neuron. And if you measure, for example, for mossy cells, the electrode tip is now here in this kind of gap between CA3 and DG and the granule cells. You see, for example, these two positions where this particular mossy cell likes to activate. So these are the place fields of this particular mossy cell they're recording from. And here, if you stick the electrode to the another place, you can record also from these granule cells, and you're going to see these other place fields here. Yeah, the colors the colors shifted because I copied from another presentation, but still. CA3 was supposed to be green, mossy cell was supposed to be red, granule cell was supposed to be blue, but everything is confusing now. But still, I think you get the idea. Mossy cells, just as a curiosity, they're called mossy because if you look them from the microscope, you're going to see this kind of, spines here that gives them this appearance of being mossy, right? Okay. So we want to do now is we want to build models that, for example, are able to learn this trajectory and replicate mouse behavior. So this is just an example. We want to make like this kind of stochastic neural network with some sort of plasticity that can kind of mimic the behavior of the mouse. We're on the way to do this. I'm going to show you some results about that. But now what we're doing is we are already printing this kind of shields to put on Arduino. We are building these Arduino robots so that we can implement this and other different types of algorithms or models for learning trajectories. But the challenge here would be, I mean, we can simulate this in the computer as I just showed you, but the challenge is how to integrate input signals, for example, infrared data from sensors, which would mimic the active sensing of the mouse, how to integrate that with the learning of the path, and so on and so forth. So this is the challenge for the robot, right? It's not necessarily designing a model that can learn a trajectory. You can do that with simulations. But for the robot, here we want to answer questions such as, how do I integrate input signals into my model so that it can learn and make what the simulation does? And we're in the process of doing that right now together with Mateus, which is a student of mine. And okay. What time is it?
You have, say, 8 minutes more.
8?
I mean, you asked me to let you know when
Right.
to go. Yeah.
So how long is the seminar? It's like 50 minutes?
It's about 1 hour time, but we should allow time for the students, for the audience to ask questions.
Right, so it's 30 minutes now, right? It's like 40, right?
It's a quarter to 11, so
Right.
finish saying 10 minutes time.
Right. So I'm gonna just go a little bit faster here then. So here is we did some recordings from mossy cells. As I told you, these are like play cells. You can insert electrodes into them in a controlled environment, in vitro, and then you record the membrane potentials, and you see, for example, that it's very, very noisy. You can apply some drugs like CNQX or PTX, and you see that PTX has no effect, but CNQX silences all the noise. So this means that the noise is coming from excitatory synapses preferentially, and it kind of makes the trace of the membrane voltage more clear. So I'm just going to skip through this part, and what I'm just going to show you tell you, instead of discussing in details, is that these mossy cells we're recording from, they are very adaptive in terms of the threshold that where the emitter spikes. So for example, if you inject this kind of current for the mossy cells, this DC current, you're going to record the spiking of the mossy cells, and you're going to see that the first spikes happens, for example, at this membrane potential, but this last spike in the series happen with a 10 millivolts at 10 millivolts higher than your initial threshold, right? So this is a signature of threshold adaptation, and it happens both as a function of the injected current magnitude and as a function of the spiking of the cell. So, and then when you release, I'm not going to give you these details, but when you release the current, you're going to see that the threshold is going to take some time to recover, and it usually is exponential, and you can fit this exponential and you find this feature, which is the characteristic recovery time, about 500 milliseconds, both for CA3 pyramidal cells and mossy cells, right? So this is the important message. We did some modeling to capture this threshold dynamics, not important right now, right? But the thing is, the model captures well all the features of the spiking adaptation, of the threshold adaptation that we have seen experimentally in mossy cells. And then we ask here is, what is the function of this adaptation? What does it do to your system? How it helps, if it does help? And then we go into the population scale, because if you want to answer a question for, say, how does the brain code information or encodes information, you have to have more than one neuron usually to do that. I mean, your brain at least does have more than one neuron to do that. And so here I just want to define what I mean by coding. If you have two situations, for example, a place field here and the mouse passes on this trajectory, or if the mouse passes on that trajectory, maybe that's what you see when you record from a particular population of neurons in the brain of these mouse, right? So maybe it has very similar temporal signatures for passing in direction number 1 or direction number 2. And so we want to be able to identify whether the mouse passed in trajectory 1 or the mouse passed in trajectory 2, right? But maybe if, if the signal generated by trajectory 1 looks like this, in terms of activations over time, and the trajectory number 2 looks like this in terms of activations over time, you'd be able to separate these trajectories by simply the number of neurons that are spiking over time, right? So this is what we call rate coding. And you have this typical stimulus response curve for rate coding, which is kind of the number of neurons on the y-axis, the input on the x-axis. And then, for every different input rate, you have an average output rate of the network. And with this kind of coding, if the inputs are in this region that I'm going to call you delta R, which is the dynamic range, you can separate them by rate coding. However, in the case of trajectories like this, it may be that the pattern of activations they generate is very similar in both passes, for both pass number 1, pass number 2. And so this type of situation where the trajectories would elicit very similar spiking patterns would not be able to be separated by rate coding, because they would live here in this region where the output rate of the network is stagnant or is flattened, right? So the question is how to code for stimuli in this region of the spectrum which cannot be captured by rate coding. So this is the question. Right, so we have this kind of recurrent neural network of noisy neurons and adaptive thresholds. We can turn on and off the adaptive threshold. What we do is that we inject some input rate into the network. We measure the output rate of the network, but we also measure the variance of the input rate, which is connected to the, variance. The variance of the input rate is also connected to the number of patterns it can generate because of the entropy. And just to show you quickly the result, we can control for the adaptation to be on or off. This is the model, but it's, not important. Here is the stimulus response of the network. And you see on the same stimulus response curve that I showed you before, on the y-axis, the fire rate of the network, on the x-axis, the input rate. And you can see that depending on the recovery time of the threshold, which is something that we did measure experimentally for mossy cells and CA3 pyramidal neurons. So that's the control parameter we're interested in. And you can see that as the input rate goes down, the output rate flattens out. And this is what you expect. And because this networks contain is made of adaptive neuron with threshold adaptation, you can control for adaptive adaptation time scale. And you see that the longer the adaptation time scale, the further the curve goes here. So the better it is at capturing weaker and weaker stimuli, right? So weaker stimuli here where the input rate is quite weak. And you see that, for example, for tau equals 10,000 milliseconds, it captures kind of almost 10 to the minus 3 input rates. Okay, so this has nothing new compared to what we already knew. However, and this only happens with threshold adaptation, now you can plot the variance of the fire rate for the same values of the recovery time. And you can see, if you line them up, so these circles here, they are for tau equals 100, which is the same as the circles up there. And you can see that every time the fire rate flattens out, the variance increases. And this happens for tau equals 1000 milliseconds. The fire rate flattens out, the variance increases. Fire rate flattens out, the variance increases. Fire rate flattens out, the variance increases. So here we're unveiling this kind of variance coding, where when rate coding stops working, the variance coding kicks in and is able to capture weaker stimuli than you would be able to capture with pure rate coding. And we hypothesized that this is the strategy or the function of having threshold adaptation in the hilus. So all these neurons that I talked to you about in the hilus, the CA3 cells, granule cells, mossy cells, they're all having threshold adaptation.
Mauricio, 2 minutes.
5?
10 minutes to go, yeah, to finish. So if you
Okay.
could say wrap up.
Yeah, I'll wrap up. So yeah, basically this is the message that I wanted to convey. You have rate coding capturing weak stimuli, and this rate coding is capturing stimuli for this time scale that we recorded experimentally. And if you just, to finish, if you plot, for example, the mutual information between input and output and the entropy, of these signals, you're going to see that there's this maximum entropy and maximum mutual information for the recovery timescale of about 1 second, which is 1,000 milliseconds, whereas the dynamic range of the network kind of saturates, increases, it enhances with the recovery timescale, but it kind of saturates as well. So there's this optimal processing of information here happening for 10 to the 3 millisecond recovery timescale of the threshold, where you have kind of good dynamic range and optimal coding from variance. So this is the message, and I would like to thank you for listening, and I'm sorry for my rusty English and for my rush. And here are the links and collaborators with whom I work with right now. And thank you, the funders.
Thank you, Mauricio, for this nice talk. So now we have, say, about 10 minutes for questions. I have already one here, which was sent by Peter Rodriguez. So, Peter, are you there? Could you?
Yes, hi, I'm here.
Okay, yeah. So
Yeah.
if you could start.
For sure. Well, first of all, good talk, and your English is excellent. Don't say that. My question is kind of more from an experimentalist background, because that's what I do. I'm just wondering how you verify with these measures. I think it's pretty difficult to do, but that, when you mark a place cell that corresponds to a certain place area, how does someone verify that that neuron wasn't spontaneously active at that time? But is there some way to do kind of a repeated measure that specific neuron adjusts to that place field sort of over and over again, or maybe can even get strengthened with time?
Right. So what you do there is you have this kind of basal fire rate of the cell, and you do some averaging of the fire rate, and you see that there's this quite clear increase in the average fire rate at that particular position. So that's basically how you do. So you have the spontaneous activation, and then you clearly see that when it goes through that particular spot, it increases dramatically. So that's how you do. I mean, of course, there are some experimental parameters that you have to kind of infer and fit and do some knob twisting, but still you're able to capture that usually by mean fire rate increase of that particular cell.
So you see that mean firing rate increase as an animal trains? Like, can you record from the same set of neurons over time?
Right. So that we're doing right now, for example, we're repeating all the experiments that I showed you with the mice here, learning, and we're recording from these cells, and that's what we're doing right now. But the experiments that I showed you from Goodsmith, where they see these place fields, these are on freely behaving mice. So they're not doing any particular task. They're just hanging out in the arena, and then you're recording and seeing the place fields. However, this is a good question, because during the learning, we can see this kind of shifting of place fields, and sometimes the mouse learns more about the environment, and then the place field can shift towards near the target. And this is something that we discuss in the paper that we published, but we didn't record actually from the neurons during the experiments I just showed you. So we didn't have the evidence for that, but now we're doing these experiments and we're starting to see these things. But I think it's very incipient at the moment, because Kelly, who's responsible for these experiments, is now a mother, so she's in maternity leave. So it's taking a while.
I understand. Well, thank you. I appreciate it.
Yeah, thanks, Peter. I remember you from LASCON.
That's it. Yep. I'm still around.
Cool.
Any other questions?
I can ask a question, maybe. So thanks for the talk. So at the beginning of the talk, when you move the exit for the mice, it's not clear to me that the mice know that it's being moved, because the setup is so symmetric that he may just think that that he's always exiting from the same door, and that the arena has shifted. So is it possible to make landmarks that are a little bit less subtle? So, for example, suppose you put a flag on top of the hole that has food. Would he be able to go to that flag?
You mean here, right? So
Yeah, the landmarks seem to be just things on the wall, which for me is a little bit subtle.
yeah. So I mean, if you put a flag on the hole, for sure it's gonna go there.
Okay, but then so it knows about landmarks. So it's
Yeah,
not
for
that-
sure. The point of these experiments is not to say the mice do not use landmarks. The point of this experiment is to say that depending on the environment, they can and will ignore landmarks because it's more like this environment's more prone to, pure path integration. So they just do path integration, right? So of course, if the landmark is very pronounced and, they will and can use landmarks. So the point of this experiment is not to say the opposite. It's just to say, depending on the environment, they will choose path integration. And the second thing is, even with pure path integration, they are able to make shortcuts and build this kind of cognitive map according to this definition of cognitive map, right? So yeah, this was one of the criticisms we had during the publication of the paper is, okay, but these landmarks are on the walls, but we did this experiment with and without landmarks and everything checked out. So yeah, we also did, and I didn't mention because it ended up not being published, we put some true landmarks like so for example, this kind of rock that we put in front of the entrance, and sometimes if the mouse find this rock, it was supposed to take the right. If it finds like a square or whatever, an L or Q, whatever other form, it's supposed to take the left. And it never worked. I mean, because this environment is kind of very big compared to the mouse size, that's our hypothesis, is that the mice are just ignoring landmarks and just going with path integration. That's what we found.
But do you think it recognizes that it's leaving from a different door? Does it
No,
know
it's
that
it's supposed not to actually.
I see.
Because if it was supposed to recognize, then I think the point of the experiment would be ruined. And so that's why everything is symmetric. Because
Okay.
you
I
know,
understand.
yeah, if you had any other kind of cue in the environment to tell them you enter from a different place, right? So then we would not be seeing evidence of path integration. We would just be seeing evidence of landmarks.
Okay. Yeah, I would be confused in that setup too, not just a mouse. But okay, thanks.
Yeah, everyone would, but the point is that eventually, even us humans would be able to
I
make the
understand.
same prediction that the mice did, right?
Yeah. Okay, thanks very much. It was a nice talk.
Thank you.
Other questions?
May I?
Yeah, of course.
Hi. Thank you. Mauricio, great talk. Thank you very much for this. I have an ignorant question, which is how much diversity do you find in the behavior? So how robust is the behavior across mice? Did you find mice that were learned faster or slower, or that did use the landmarks, but it was just one or two mice? Can you comment on that, please?
Yeah. I mean, we found that the mice are quite smart. They are way, way smarter than we expected. And to be honest, this kind of static entrance experiment that I showed you, we did with over 70 mice. Like we did with a lot of, I mean, Kelly did, poor Kelly. She had a lot of time in the lab to do all the experiments. And and the static entrance protocol was never a problem. I mean, every time with every condition, whatever condition you think about, Kelly did it and the mice repeated the same thing. The only thing that was a little bit kind of more delicate in terms of finding is this two target experiment where the mice have to take the shortcut here. And I'm just gonna give you the video just to.
So in that case, you had some mice that would learn and some mice that wouldn't.
Yeah. So as I told you, it's not that they wouldn't learn, but if you check their trajectories, we put out the trajectory examples in the paper, but here, some mice, for example, as I told you, they go to B and then back to entrance and go to A, but the majority of mice do this that I showed you. Majority, for example, in terms of percentage, 5 out of 8 mice would do this. Like they go to B and then to A. But you have seen examples, and this is in the paper, for example, where the mouse goes to the right, but it never quite finds B, and then it comes back to the wall, goes back to the entrance, but then does this whole turn here, and then goes to B from there. And then it goes to A. So, you know, we can see that these mice are kind of very smart, because even though it didn't know it had to go right because B was there, but it didn't find anything. It does a whole turn and comes back to B and then goes to A. So we wonder what's in the mind of this guy, because it clearly knows what it's doing. And, you know, so we see different kinds of examples. And it's quite hard to kind of filter out what they really know or don't know because of the open environment of the arena. If the arena was more like a kind of a team maze or whatever, it would be way easier to try and understand what the mice are doing. But our point is actually to make the environment open so that we give them freedom and we kind of observe their more natural behavior. You know what I mean?
No, I totally agree. I think it's beautiful. We also work on open field and field behavior, and this is one of the problems. But you were talking before about active sensing, right? So in this open field experiments, then engagement becomes a huge question, because at some point they might just not be doing the experiment and then they log back in, and so engagement really triggers different behaviors and also different neuronal patterns. So I found it really interesting when you said that engagement could be measured in how many holes are they checking.
Yeah.
Maybe when they're walking around and they don't fit what you expect, is because they're not engaged in the experiment. So you could check with hole engagement or something like that, right?
Yeah. So this one very important feature of the experiment is when we, kind of had this insight of measuring hole checks, because at some point we just had trajectories and we were just analyzing trajectories and the kinematics, you know, distance, time, velocity, and things like that. But then at some point we had this kind of insight, well, maybe. If you look at the videos, you can clearly see that, you know, they stop, they check. And then, you know, like I'm showing you right now, they stop and they check. So, and you have a very clear signature in the velocity, acceleration, and trajectory of the mice. So you can kind of use this kinematics and do some filtering of the data to be able to extract exactly when the mouse is engaging in this kind of active sensing and checking the environment.
Exactly, but you haven't done it yet, right?
Huh?
You haven't done it yet, right?
Well, we did, yeah. We hadn't
okay.
done it before, but we're doing it, you know.
Cool.
Yeah.
Beautiful. Thank you very much.
Thank you.
I think we can have still time for one more question, short question, if anyone in the audience. I have one then. Mauricio, it's well known that rodents have an innate preference for vertical surfaces like walls. So in that naive model that you presented to us, the random walk model, have you tried increasing the probability of stepping towards the wall instead of going towards the center of the arena?
Yeah,
That
this is
probably wouldn't change very much, but probably would increase the time, the learning time. I don't know.
interesting, and actually something that you see in experiments as well is that even in the first video that I showed you, where the mouse kind of goes on the wall and stays on the wall a little bit, and then it goes to the center, it's a trend that you see in all experiments here in open fields. It's a very natural behavior for them to avoid predators and stuff like that. When you go to the random walk model, this is something that naturally emerges from the modeling. Here, I'm just going to show you. You can see here that there's a natural, in the beginning, there's a natural trend of probabilities being high at the edges. And this happens because at the edges of the arena, you have less sites to choose from in your random walk, right? So, for example, if you are at the corner, you have only 2 sites to choose from, right? Instead of when you are in the middle, you have 4 sites to choose from. So when you are in the border and the walker is in the border, it kind of has a natural trend to stay near the border, but then it kind of can get out of the border. So So yeah, it's something that we naturally see in this random walk scheme, although I don't know if, you know, for example, maybe the wall could be used as a cue in actual experiments for mice, you know, because we clearly see in some trajectories the mouse stick to the walls, and instead of going straight to the food, they go through the wall and then they go to the food. You know, it's something that we see in many trials. So, but it's not something that the model would be able to reproduce, I guess, you know, at least in the first glance.
Yeah, that's what I asked. Probably you have to add by hand
A
what
bias.
function did, because it's innate for them. It's not something that they learn. Of course,
Yeah.
you'd be able to model that with your function,
Yeah.
your learning function. But then if you just add this bias towards the walls, maybe
And
it would.
also there is another bias that Kelly, who's the specialist in this kind of experiment, she was telling me that mice don't usually go back. I mean, they go forward and to the sides, but they don't necessarily do this, right? They're not doing this. They, as the walker would do, because it's just 1/4 going forward, 1/4 going backwards. So the walker can do this, but the mouse usually either goes there, left, right. It can take a turn, but it not necessarily goes back at the particular site, you know? So it's something that the model can be improved on.
Okay, thank you, Mauricio.
Thank
I
you.
think, yeah, I think we have expired the time now. It's about 10 past 11. So let's thank you again. If you could say, clap hands or whatever.
Yeah. Thanks.
Thank
So
you.
our next meeting will be, say, in a month time, I believe. No? Yeah.
Yeah,
Announced.
hopefully on a Friday.
Okay.
Bye-bye.
Right. So bye, everyone.
Thank you.
Mauricio again.
Thanks.
Thanks to the audience for coming.